All right. Welcome, Forsyth County Beekeepers Association members. Pleased to have you here. I'll be conducting the meeting tonight. I'm Sarah Devine, the past president and current programs director. Burl has an annual event that he has to go to, so he won't be coming to conduct the meeting today. So as I said, welcome, very pleased to see everyone. I wanted to let you know that we will be giving out door prizes at the end of the meeting, so stay to the end if you want door prizes. Some old business, we're going to have a brief business meeting and then we're going to jump right into our speaker tonight. So our old business, we had a very nice socially distanced September meeting, our only meeting since the COVID-19 came by that was in person. Thanks to everyone for driving all the way out to the Woosley Farm event barn. We enjoyed having Michael Bush talk about the winterization of bees. It was very handy and very timely. I'd like to go, Steve, are you ready for
yes.
for treasurer's report? Okay, yep. Let's go ahead and do that.
So starting September 2020, club funds were a total of $12,683.13. This is with BB&T at $12,647.83 and petty cash at $35.30. We had a net change of $3,320.64 for new balance of $9,327.19 in BB&T. Petty cash remains at $35.30, so we enter the month of October with a total of $9,362.49. Thank you, guys.
What was it, 9,300 what?
$9,362 and 49 cents.
Okay. All right. Thank you very much.
Of course.
A couple of programs that we had in September. We had the Feed the Bees program, which we had our fourth Feed the Bees giveaway, and that went very successfully. Thanks everyone for getting involved in that, making sure that our bees have enough stores to get through the winter. And we had our linden tree sale. Linden trees are also known as basswood, and they bloom rather late in the year, roughly around the time that sourwood blooms. So we're trying to get a few of those in the area. We sold 100 of those, and that went really well. Everyone was cooperative, and it was quite nice to have. New business. Usually we have fairs going on at this time. We have the Winston-Salem Carolina Classic, which used to be the Dixie Classic Fair. It's having a drive-through fair October the 1st through the 4th, so you still have one more day to go. The State Fair, I understand, has been canceled, and I haven't heard anything about the other fairs, so they're probably canceled. Our next meeting is going to be on November the 7th. Dr. Jennifer Berry from the University of Georgia will be speaking, and the topic has not been determined yet. The last thing that I'd like before we get started with the speaker is we have some open slots to run the Forsyth County Beekeepers next year. The president will be Burl Childers on the slate if you vote for it. Vice President David Link, Treasurer Steve Mutai, librarian will be Jeremy Tyson, who's moving over from the recording secretary to the librarian position, which he's wanted to do. June Hartness is going to do the bee school again. Thank you very much. I'll do the programs again. And so we have three things that we need. We need someone to be the recording secretary, which is fairly straightforward. It's keeping a notes of what goes on in the meeting. Somebody to do the newsletter once a month, about a week before the meeting, and to monitor our Facebook page. And then we also have membership secretary that we need. That's pretty heavy at the end of the year, the beginning of the year, mostly around bee school, which is in February, March. So those are the busy times for membership secretary. All right, please get in touch with me or any officer if you would like to help to run the bee club. All right. Tonight we have a speaker who's very well traveled and has agreed to meet with us. His name is Dr. ESMAEIL AMIRI, and he's going to talk with us about queen and viruses, transmission routes and infection consequences. Dr. AMIRI used to be a migratory professional beekeeper in Iran along with his undergraduate studies. So he has some good practical knowledge as well as academics. He then traveled to several European countries, including Germany, Sweden, and the Netherlands to broaden his knowledge about bees. Furthermore, he earned his PhD from Aarhus University in Denmark. He then traveled to North Carolina to work with our North Carolina big names, Dr. Olaf Ruppel and Dr. David Tarpey, investigating queen virus infection and consequences to the next generation. Dr. AMIRI has been contributing to the field of honeybee science since 2014. He's published several scientific articles, which is good for someone who's in academia. His academic interests lie in the interaction of the fields of honeybee health and virology, specifically virus-host interactions and virus transmission pathways, with the aim to identify alternative ways to combat viral diseases in honeybees. All right, Dr. AMIRI, if you would go ahead and start your talk, we'd appreciate it, and welcome.
Thank you, Sarah. Hello, everybody, and I'm very pleased to join you tonight talking about part of my research results. I'm not going deep to data and all those graphs make you more confused. I'm presenting something that I enjoy and you will enjoy as well. So I have been working with honeybees and honeybee queens for a couple of years. And I'm sure that you know all about the queens, and we all know the queen for laying eggs, but it's interesting to know that before queen lays eggs, she needs to show her royalty in the colony for the bees to respect her. And that's why she is releasing pheromones and attracting bees to treat her nicely and keep the colony as cohesion and working toward one aim, the colony's success. And that's why in this regard, if she is combating with the other mates, with other queens or other workers that are laying eggs, she is able to attract bees and bees will protect her. And as you see, like even during the pupae, if she is successful in that regard, worker bees will respect her and keep her safe. Or even after emerging, you see this picture in the right-hand side. This is not something scientific, even though recently people are interested on temperature on queens and the effect of high or low temperature on queens. But as you see here, it just shows that how worker bees are protecting queens in the colony and keep the cool temperature for the queen. So beyond that, after she is successful to be head of the colony, she is able to lay eggs. Otherwise, if she is not successful to attract the bees and show her royalty, she will not she will not have chance to lay eggs. That's why, first of all, she needs to show that she is a real queen and can lead the colony. So she is very important for the colony in that regard. Then it's interesting to know that, and you know all that, an egg can go either to worker bees or going to queen. And in each of these levels, from egg to adult queen, she is facing infection and facing the way that she receives viruses to be infected. As one colony, as the queen in the colony, we know that the colony is under different stressors, and as like the queen as well. Either it's agrochemicals that coming from outside or inside the colony, nutrition, pollen, nectar, or other pests and pathogens. And here I am mostly focusing on viruses. And we know like beekeepers and temperature and environmental effect can impact the colony and as well as the queen. With viruses, we know viruses are in different categories, but I'm not going too deep in that categories, but we all know that viruses are impacting negatively worker bees and the queen and as total as colony. What I'm talking today is about deformed wing virus, Israeli acute bee paralysis virus, chronic bee paralysis virus, a little bit of black queen cell virus, and show you how these viruses can transmit or have consequences on the queen health. Let's start with deformed wing virus. Deformed wing virus, I'm sure that all of you have seen the bees with symptom of deformed wing virus. Deformed wing virus in association with Varroa mite can cause individual death or abnormality like the picture above, or during the winter killing the colony. But it's mostly during the winter. I mean, this deformed wing virus, this virus can be found in all castes, queen, worker bees and drones, can be transmitted horizontally from worker bee to worker bee, from worker bee to pupae to larvae, or worker bee to the queen, or worker bee to drones, or through Varroa mite, because Varroa mite can just suck up the hemolymph of one bee and then jump to the larvae or another worker bee, or vertically transmitted, coming from when we are talking about vertically, we are talking about virus transmit from queen to the eggs. And as I said, it's mostly linked with the colony losses. But we should know that these colony loss that I'm talking about, we should be sure that these colony loss, that most of you see it in the winter, or you will notice it in the beginning of spring. It is because of Varroa mite and the virus, that you see how many Varroa mite you can see in one cell. But most of the time I have seen beekeepers that showing or complaining about death of the colony or colony loss like this one. But to my understanding, and specifically working in North Carolina for 5 years, these colony did not die because of Varroa mite. These colony died because of less food availability. Because as you see here in North Carolina, we do not have very cold winter. We have a mild winter. And that's why the colony doesn't go to the real winter colony or cluster, winter cluster. And because of that, bees are active and consuming so much honey. And most of the time you see colony is alive until January or a little bit after January, February, and then dies afterward. And it's because after January, February, January, the temperature, specifically after January, the temperature rise, and then bees will eat more and they don't have enough honey. Queens start laying eggs because more nectar, more pollen is coming. But then in March, end of February, March, or beginning of April, you see more activity, but not enough nectar, plus fluctuation of the temperature. that cause diseases and colony death. These colonies don't die because of Varroa mite or deformed wing virus. They die because of less food in the colony. If you want to know, because most of the beekeepers are complaining about, okay, we did not treat, or whatever we did treat with for Varroa mite, it didn't help. And our colonies died because of diseases, Varroa mite and deformed wing virus. And actually this is not true, because easily you can test your colony. If you have a colony, dead colony, and you break down the frame, and you see inside the cells, and you see these white things full of, I mean, most of the cells are having these white things. They are defecation of Varroa mite. And for such a this colony that you see out of, like, let's say 10 cells, 7 of them having this white feces of Varroa feces, you can see, okay, this colony died because of Varroa mite and diseases. Otherwise, if you don't see such a this phenomena, you cannot say, oh, our colony died because of Varroa mite and diseases. Maybe your colony died because of less food availability in the colony. So the second virus is chronic bee paralysis virus. It's a virus, at the beginning was thought that it's not in America, but then it was found in America, and recently it increased the prevalence. Most beekeepers are complaining about this disease. Normally, disease is can be seen in worker bees. It has 2 types of symptoms. One type, as you see in the picture, bees will turn black and hairless. They become like a hairless and black and greasy. And these bees are attacked by other bees. And they can, still with these symptoms, these bees can fly for a few days, but finally they die. The other symptom is bloated abdomen, and bees cannot fly, and they die gradually. And as you see in the bottom part, colony in 1 or 2 weeks, you see lots of dead bees are removing by the other bees, and in front of the colony, bunch of worker bees, still they are, or sometimes they are huddling in front of the colony. And if you open the colony, you see these infected bees coming on top of the frames and start just shaking, abnormal shaking. And mostly this virus cause death of the colony in the summer. Another disease is Israeli acute bee paralysis virus. This disease is one of the diseases or viruses connected to colony collapse disorder, and associated with Varroa mite as well. You see these are infected, colony is like in few days, colony is dead. And you see the symptom, how bees cannot fly, the 2 legs, 2 hind legs or back legs are not functioning, and bees have difficulty to walk. Like very quickly the colony will die. But then we should all know that viruses are transmitting in different pathways. One pathway that I will talk about, it's how queen is infected by drones through mating, or how if you have even like a beekeeping thing, if you have one colony infected, let's say you did not treat it against the Varroa mite, and then you exchange the frame and such these things, and then you can just transmit the infection from one colony to another colony. But inside the colony, as you see up here, bees, worker bees, as I said, worker bees, if they are infected, are passing the virus through feeding or food exchange to larvae and pupae, to larvae, or among themselves, or virus can transmit through Varroa mite, transmitting virus through to the queen, or if you are a very careful beekeeper, but still you have a neighbor that he or she is not careful about the colonies, because bees are they are collecting nectar from the same habitat. So the neighbor beekeeper is not careful, and his bees are infected either with Varroa mite, either with viruses, and through the same habitat or same flowers, bees can get infected as well. So your bees become infected in that regard. Bees can be infected while you are catching the swarm. I mean, if you are catching the infected swarm, you are catching, if you are catching the swarm, sometimes you are catching infected swarm, or through rubbing. So many times we have colonies, and colony is not a big colony. We just open up the colony and let the other bees in the apiary just to rub this colony. But we need to think at the beginning why this colony did not grow, and this colony could not increase the population, because there is a disease. And if you open the hive, or if you just leave the frames over there outside, and other bees are attacking this colony and robbing this colony, it means that they are not only taking the honey to their own colony, but they are taking diseases as well. And many times, even like during the fall time, you know, from the beekeeping point of view, people are saying, okay, maybe we need to combine the colonies, like combine the small colonies with big colonies. Sometimes, you know, the colony is not growing because the queen is not good, but still the bees are healthy. But sometimes the colony, even though the queen is good, the colony is not growing because the colony is dead, is infected either with viruses or bacteria or other diseases. And that's why the recommendation is, first of all, we need, before combining the colonies, we need to inspect the colony, the weak colony, and see if there is no disease, then combine them. If you see the colony is infected by whatever pathogen, you should not combine the colony, because when you add the small colony in the big colony, you are increasing the disease in the big colony. So it means that you are adding diseases to your own colony, and that's why it makes it problematic. I have, if I want to just talk, I have two papers. They are open access, and about honeybee queens and virus infection, and honeybee and queen quality and impact of honeybee diseases. I discussed about different diseases with the queen and queen quality. If you are interested, you can just download and read them, but I will just explain how one queen can get infection through the life. Let's say start from the egg, because one queen starts from the egg. This egg can be infected from her mother. We test that. We test how eggs become infected from the infected mother. We had 5 queens, queen 1 to 5. We wanted to test how virus, here is the deformed wing virus, transmit from mother queen to next generation. We had these queens tested, and we knew that they are infected with virus, with the deformed wing virus. Then we provide these queens with worker combs and drone combs. Then queen laid worker eggs and drone eggs in those combs. We collected eggs from worker eggs and drone eggs, 2 batches of drone eggs. With one batch, we did not do anything. With the second batch, we surface sterilized the eggs, and we wanted to see how virus transmit. And after washing and quantify the virus, we found out the worker eggs, because, you know, that was the idea, that maybe virus is transmitting through the sperm, or virus just transmitting from the mother, or how virus is transmitting. Then when we had worker eggs, we wanted to see, is it the sperm infecting the worker bees, the eggs, or it's the queen mother, mother queen is infecting the eggs. That's why we collected worker eggs. And we found out the quantity of viruses for each of the queens, the eggs from the queens. And then we tested drone eggs, and we noticed that the same level of virus are in the drone eggs as well. After we washed the eggs, we noticed that the virus level dropped down like dramatically. And the conclusion is that the queen can transmit the virus to the next generation, but the virus mostly stayed in outside of the eggshell. It's not less inside, but mostly outside of the eggshell. We don't know what is the virulence and pathogenicity of these virus. We have a project is running by now, but at least we got to know that this virus is transmitting from the egg, but mostly outside of the eggshell. So that is one way that virus can transmit from mother to next generation. And if either of these eggs become queen, it's possible that these queen become infected from their mother. In the same time, while developing and worker bees are providing with royal jelly, it is possible that these workers are infected. And as in many times, the virus, if we imagine ourselves as a virus, we know that if virus kill the individual, actually it's killing itself as well. But virus would like to be spread from individual to individual. And that's why, and then the virus tries to find a way to be able to transmit. In many viruses, they are either transmitting through, as I said, Varroa mite or feces, but virus can reach the hypopharyngeal glands and come to the royal jelly and infect the next generation, either through larvae, like infect the worker larvae or queen larvae. So if there is big amount of virus in royal jelly, it is possible that the queen larvae become infected. And many times if the queen is infected, she will die. The queen larvae will die, and worker bees will remove her. Many times we have noticed that viruses are in the wax that are around the queen. And because this queen has very fragile, let's say a skin, viruses can transmit to the body of the queen pupae and make the queen infected. The viruses, as I said, they are transmitting in different ways, and I have tested different ways to find out about that. Deformed wing virus, which I'm going to talk about it, I will talk about details about that through mating or through feeding. In this experiment, we kept the queens, we infected the bees and kept them in one side of the mesh. And then we kept the queen in another side of the mesh. So, and we did not provide any food for the queen. And queen always was sticking to the mesh and asking for food. And then these worker bees were around like the mesh and feeding the queen, and not only feeding the queen, but transmitting the virus to the queen. So that was like, we try to understand the pathway of how virus transmit to the next generation. Or with the chronic bee paralysis virus that you see it here, I will show you a little movie later on. Queen get infected, she gets infection through body contact. This virus is specific, and when the bees are, so many bees are in the colony, they break down the hair of each other, and virus can transmit to the body of either worker bees or drones. So let's talk about the deformed wing virus, which I showed you a picture. And the idea came in a way that, because, you know, like queens are flying out of colony and get mated. It has been a mystery for many, many years. And it was always questioned that why she is flying out and drones are flying out too to be mated there. And one idea is because when she is flying out, sick drones cannot reach drone congregation area, and mostly healthy drones reach there and are able to mate with the queen. The other idea was, well, when she is flying out, she will recognize which drone is infected or which one is healthy, and she is able to select which one she needs to mate with. And if those are not working, either these drones are able to fly, sick drones are able to fly, or queen is not able to recognize the infected drones and she mate with these infected drones, maybe there is a possibility because all these sperms are accumulating in the spermatheca. And the idea is, okay, there is a mechanism in the body of the queen or a spermatheca that doesn't allow the virus transmit to the rest of the body of the queen. And we wanted to test all these three questions. What we did, we used the Danish, because I did my PhD in Denmark, we used this Danish mating system. And you know, Denmark is like a small country, but it's 3 islands, one big island that you see here, the other island is small in the middle, and the other like a little bit bigger island in the corner. But around these 3 big islands, there are small islands that people are living there and they are doing agricultural activities. But beekeepers use this system and have their queens there, virgin queens, and have their selected colonies to produce drones and have them all there. And it's like a controlled mating for their system. And it's working fine, very fine. And that's why they are very successful of selection, because when you are trying to improve the trait, let's say production trait or health trait, you need first of all to recognize the colonies, the right colonies. And after knowing or identify the right colonies, you need to find a way that these queens from these colonies, selected colonies, be able to mate with the right genetic background of drones. In our system in America or other countries which we don't have such these islands, most of the beekeepers or queen breeders are getting advantage of artificial insemination. But with artificial insemination, how many queens in a year you can inseminate? I'm not sure if people can inseminate more than 1,000. But with this island mating, everything is just free, and they can just make these queens maybe more than 10,000 queens in a year, in one summer. So it's a good system over there. They utilize this advantage that they have. So that's why we use this system, and we had colonies in one island called Nexolo, and another apiary we had kind of close to the slag, like a little village called Flakebia. And in this little island, and let's just go forward and then I will explain. We had a mother queen, and we did test this mother queen and the colony for being led to know that this queen is infected or not infected. After knowing that the mother queen is not infected and the bees around her are not infected, we asked the queen breeder to graft for us. After grafting and having them all in one nurse colony, we tested some of the nurse colony bees to know that they are infected or not. Plus, we took 3 days old larvae with royal jelly to test the colony and larvae, are they infected or not infected. The rest of colonies, we did let them to emerge. We took 10 queens afterward, like right after emerging. And we had 12 of these queens in a small nukes and moved them to that island, separate island, and provide the healthy colonies to produce drones. We took 38 of the colonies to the other island. Let me show you. We had 12 queens in this little island with the healthy drone colonies. With 38 colonies, we had them in this big island and provide these virgin queens in a small, of course, we introduced them to the small nukes. And because they are ready to mate, we provide that colonies or that queens with colonies that were not treated against Varroa mite. And that's why the Varroa mite infestation was high. Level of viruses was high as well, because we wanted to see if these queens are infected or not, become infected during mating. So that's why we had them. And then because we wanted to control the environment, we didn't let the 8 of these queens to mate. We treated them with CO2 2 times and had them in the colony and had queen excluder in front. We did not let these queens fly for mating. But for 30 of these queens, we did let them to go mating, and then test them. Gradually we test them. We did test them. The first group with royal jelly, and here you see, when you see different colors, if we found no virus at all, we show it with a blue one. If we see some level of like a small amount of virus in that samples, we show them with a low, let's say green, and if medium or high level infection with orange or red color. With royal jelly, we did test all of the 15 royal jelly samples. We did not detect any virus. With larvae, we detected a little bit, but still nothing to be worried about. When we had newly emerged queens, we did dissect them to 3 parts: head, thorax, and abdomen. And we noticed some of the queens showing virus, but still not in high level. They come up to medium level, only two samples, but not all of them. With queens in Nextolo that we took them, after bringing them back, we dissected them. And as you see, we cut the abdomen, took out the ovaries, as you see here, and then took out the spermatica, took out the sperm out of the spermatica, and test all these samples. Let's say we had head, thorax, abdomen, ovaries, spermatica, and sperm. Checking them for virus detection, we see, okay, now we see some virus are showing up, but still there is no way to be worried about. The level of virus is not that high. But what we did with the queens in the infected colonies, how we controlled those colonies, let me see. So we had all these queens in the mating nukes, and as soon as the queen is flying, is coming in front of the queen excluder, we did let her to fly out and establish the queen excluder again. And after she is mated, she is coming back. She's coming back, and you see these are directing her to her colony. Slowly bees are coming back, and you see more action bees are doing and more pheromone, and the queen is showing up, coming back. When she is coming back, she is coming with a little white thing in the back. And this is the, let's say, endophallus or penis of the last drone that she was mated with. So she brought that sample for us. And it was the time that we wanted to have this sample and see whom she was mated with. Because that drone is not able, is dead, already dead, and is not able to mate with any more queen. So that's why we took these endophallus as one sample, and we took it out, as you see here, as for one sample. And let the queen go in to just lay eggs. And she's happy. And then just establish the queen excluder and do it over and over. For a few days, we just walking through all these nukes. And every time we see the queen in the back of the queen excluder, let her fly out and then establish the queen excluder. And after coming back, let's see if she's bringing any sample for us or not. If bringing, that is good. If not, then that's one sample we had from her already. So then I told you that we had queens that they were like did not mate. They were in the colony. We treated them 2 times with CO2. And we did test the different part of the body: head, thorax, abdomen, ovaries, and spermatica. Because they did not mate, they didn't have any sperm. So that's why they didn't have a sperm, and we did test them. And only a few of them, like few tissues were infected, but not much. It is like not a big worry. When we tested these samples that we collected with this endophallus, let's say, you see there are some white rows that they are representing for a queen. We didn't get any sample from these queens. But for other queens, for example, for this queen, we got only 1 sample. And that's why we did test it for the virus. For some samples here, let's say here, we got 2 samples. One day first and day second. What does it mean? It means that queen flew 1 time today, and then she got mated with few drones, and she brought 1 sample back home, and we took that sample. Again, tomorrow, she felt that it's not enough. She needs to mate more. She flew again, and then when she came back, she brought another sample for us. So the last 2 drones that mated with her. And then we did test them for the virus quantity. And we found out most of these drones were infected. And how about the queens? How queens become infected? You see how these queens in the same area, they did not show any like high level of virus. From 30 queens that they were allowed to mate, 6 of these queens showed high level of infection. 1 queen died. And if you check them with the drones, we see that these drones were mostly infected. And that's why these queens got infections through drones. The other 7 or 8 queens here, they showed medium level of virus as well. So they were infected, even though we see less of them are mated with drone that were infected, but we need to remember that queens are mating with 12 to 15 or 20 drones. So it is possible that these drones were not infected. The samples they brought back, they were not infected, but it's possible that they were mated with other drones that were infected, and that's why they become infected. Some queens, like 6 or 7 queens, did not show any like high level of virus. We don't know. Even like one of the queens here, she mated with high level of with the drone with high level of virus, but she did not show any virus in the spermatica or sperm. We don't know what is the story of that. But and that is interesting. At least these groups, maybe genetically they are not susceptible to virus. They are tolerant to virus, or virus did not get chance to replicate in their bodies. We don't know the story yet. So this is how one queen can get infection through mating. Let's say talk about, and this is different ways. I mean, maybe I needed to add one slide here showing how infection can come through feeding when she is receiving food from worker bees, or mating, or body contact. But you see queens can get infection in different stage of development. Even during the pupae or larvae or pupae, they can be infected. If they, during the pupae, the larvae, they are infected, I'm sure you have seen such that queen cells that were like cannibalized by worker bees and removed from the colonies. Or later on, when they become like a pupae, still in the beginning of pupae, end of larvae, they become infected and the cell chewed by worker bees and removed. As you see here, black queen cell virus, how queens are infected by black queen cell virus, and they will not develop, they become like a queen. And worker bees notice that, and that's why they chew the cell and remove the larvae or pupae early later in the larvae or beginning of the pupae. Sometimes we have deforming virus. I mean, I did test, I mean, this is what I test. This is not most, like a beekeepers or queen breeders, let's see such a these symptoms. We wanted to test because Varroa mite cannot develop in the queen cell for different reasons. First of all, Varroa mite doesn't like to go to the queen cells and infest the queen. The second of all, because the developmental stage of the queen is too short. It's only 16 or to 18 days. But the Varroa mite needs 20 to 24 days to be able to reproduce and have next generation. So that's why queen is not the right host for Varroa mite. But we did test that, and we wanted to see, because we are a scientist and we want to see what is the symptom when we introduce Varroa mite. And you see, we introduced the Varroa mite to the queen cells, and only like 2 or 3 or more Varroa mite we introduced to the queen cells, and you see the symptom. Queens emerge by deformed wings. I did talk about chronic bee paralysis virus. We wanted even, you know, in the colony, in a big colony with a couple of thousand worker bees and only one queen, you cannot see the symptom of the queen when she is infected. Most of the times, beekeepers see dead queen in front of the colony, or maybe you will not notice it at all. And that's why most of the experiments are doing in the lab to see what are the symptoms of the queen when she is infected with viruses. And that's why this is chronic bee paralysis virus that I said it mostly transmits through body contact. And in here, we just shave the hair and apply the virus, just add the virus in top of the thorax and let the virus just to absorb by the queen. And then after 7 to 9 days, you see the symptom. The queen cannot walk. She is paralyzed because it's a chronic bee paralysis virus. It's just paralyzing of the queen, and she cannot walk. Then when you open up the abdomen, you see the abdomen is full of hemolymph. Or in a case of Israeli acute bee paralysis virus, we did the same thing. We infected the queens, and you see after 24 hours, this is the symptom. Queen is like paralyzed, and because most of these viruses are infecting the nervous system of the queen or worker bees. And these are the symptoms. I showed you the symptom for worker bees earlier, but this is how we got it for the queens. And finally, if you find out the queen, infected queens, and you see like it's in, worker bees are gathering around the queen and balling her, and in, let's say, a couple of hours, they will kill her in a way, in one way or the other way. You end up having a dead queen. So let's say how we control the virus in general. It's not different from us controlling the virus. We know that when we are infected with virus, we are quarantined, we are separated and taking rest. We are washing our hands and to not transmit the virus from one person to another person. Or we use vaccine. The same system with the honeybees as well. There are different ways to control the viruses in honeybees. So if we see any symptom with the colony, we need to be able to make sure that we are separating this colony from the other colonies. We are not using the same tools. This summer I visited a beekeeper and she had a couple of colonies. One of them was showing chronic bee paralysis virus. And then she was using the same tools with the same for all colonies that she had. And suddenly she just transmitted the virus from colony to colony, and she ended up having like few colonies, all of them showing symptom. And that was like a tragedy for her as well. So that's why the thing is, if you see any symptom with a colony, maybe you should just not use the same tools for all colonies, or you should not exchange the frames between the colonies. Be careful about that, specifically with Israeli acute bee paralysis virus or with chronic bee paralysis virus, because chronic bee paralysis virus is increasing. As I have heard of many cases this summer. So you need to be careful about that. If you are producing your own queens, be sure that, and you are grafting, be sure that you are grafting from the healthiest colonies that you have. Of course, with the respect of those traits that you are interested to increase them in your apiary, but be sure that you are grafting and producing queens from the colonies that are healthy enough. If you are not testing them by laboratory technique, at least know the symptom of the diseases and know that, okay, this is the symptom for this disease, this is the symptom. And if you see symptoms in that colony, don't graft from that colony or don't produce any queen from that colony. Be sure that having queens, if you have a couple of colonies, having queens from different lines. In this case, you have, right now we do not have any vaccine for viruses in honeybees, but we know that different breed of or strains of bees having different level of tolerance or susceptibility to diseases. Be sure that you have queens from different lines. In this case, you have a genetic variation in your own colonies. So then if one colony is infected, the next colony is not infected as well. If you see with this, specifically with this chronic bee paralysis virus that I showed you and did talk about it, because I'm sure that it's increasing. Be sure to change. It has like different strategies to cope with the virus. First of all is, if you see more dead bees in front of your colonies and you identify it as chronic bee paralysis virus, make sure to increase more bees from other colonies to this colony and then change the genetic profile of the colony. That is one thing in the short term. In the long term, be sure that you are changing the queen or replacing the queen. Then you are introducing new genetic to your colony. Then new generation, when they emerge, they are able to cope with the virus because they come from different genetic background that possibly they are not susceptible to virus. So different ways. And then beyond all of these, be sure to treat against Varroa mite and keep the Varroa mite level down in your colony. And Varroa mite, I know that in such that time, we do not have enough time, okay? Our time is short. But as a hobby beekeeper or as like a sideliner beekeeper, what I what I see it, controlling Varroa mite is a process. It's not like an action in like a short time. It's a process. From very beginning in the spring, add a few of, when the bees are producing comb and building comb, add 1 or 2 combs of drone cells. Then workers will make it, drone cells, and queen will lay eggs, drone eggs in there, and drone larvae or pupae are more attracted by Varroa mite. And after they are capped, just remove them. In this case, you are removing part of the Varroa mite population in the colony. So from very beginning of a spring, or let's say April, May, start to control Varroa mite. If you do that, then by the end of the summer, it's like a small thing. It's not taking so long. It's a small thing. If you're controlling from beginning of the year, by the end of the year, because many beekeepers are complaining about Varroa mite. In the end of the year, you are not complaining about Varroa mite because the whole summer you took care of Varroa mite, and you are not suffering from increase of Varroa mite level, or keep some of those empty drone cell drawn comb for sometimes in June, July, or August. Time by time, you add 1 frame of those, and after the queen laid drone eggs and they are capped, you can just remove them from the colony. If you are having like chicken or so, just give them to chickens to eat those larvae or pupae. It's a good protein source for them. You see, like in this way, you are able to control the Varroa mite plus decreasing the level of virus infections in your colonies. By that, I would like just to thank all my mentors, mentees, and my lab members, beekeepers that I have been working, and all the funding agencies that funded me for these projects. I would be happy to answer your questions if you have any.
If anyone has any questions, you can type them in the chat box. And see, one from Stephen to everyone. Are you aware of any cold room facilities that may be used to overwinter colonies in stable conditions to create that winter cluster you touched on in the beginning?
Yeah, I don't know if anybody is running such these facilities here in North Carolina, but I know that in some states people are having buildings, and I think it's at the beginning such that building or a storage were built for onion or potato, just having potato or onion in there. But later on, beekeepers notice it and they say, well, they can have their own colonies for whole winter at 4 degrees centigrade or something, and not fluctuating of any temperature. But I'm not sure if anybody here in North Carolina running such that facility. But it is a good way to have such these facilities. And we had like an idea to keep our bees for some time because we are suffering as well. We, as a scientist, of course, our life is completely different than yours. You need healthy colonies, but we need infected colonies, you know, because of all this research, we need them to be infected. Even some time by time we ask beekeepers, and we go there, collect Varroa mite and bring and introduce them to our colonies. So you see, our life is scientific life is completely different from yours. But then we just opening the hive more often than you. Even up to like recently, we were working with colonies, you know, opening, every time you are opening the hive for half an hour, for 1 hour, and then you see you are introducing some stress to the colony. So our colonies normally are not healthy. You know, it's because we would like to know the symptoms, we would like to know the disease effect, we need to know about the stress effect, and that's why we just give them different stress. That is our life here. But for you, it's completely different life. You are trying to keep the colonies healthy as healthy as possible. So so this is the case. Even if it can go with the clubs, having like such that building and running by clubs and see how successful. I see it like successful because some companies are working in it. But no, I'm not aware of anybody is running such these facilities in North Carolina.
All right, yeah. And this is not a question, but Steve went on to say that that he noticed when he was beekeeping in Pennsylvania that the cold, stable winters made it easier for him to overwinter the hives so they didn't starve. And he gave a link for a bee culture article that was published on this topic in 2017. And if you can't see that, anyone, just go to the bottom of your screen and click on the chat box. Also, if you want to put any questions in the chat box, that's where you would go. And I'll make sure that everyone gets that link. Then we have another question from Sarah. What kind of hive was in the picture sent to the FCBA for their website? The frames look more square than ours.
Let me see. I have done all of this in America, in North Carolina. You are talking about this one?
Is that the one you're talking about, Sarah?
Hang on, I had to unmute myself. It was the picture that you sent me earlier that we put up on our website for the event.
Oh, yeah. That's correct.
Down beside the hive.
Yes. Yeah, that's in Denmark. That is the colonies during my PhD. It was end of my PhD, and that is in Denmark. Yeah, the colonies in Denmark. Yeah.
Okay. Does anyone else have a question? Again, type it in the chat box. Okay, that's all I'm seeing right now.
Okay.
Yep.
All right. Well, thank you very much, Dr. AMIRI, and we are going to move right on to the door prizes.
Okay. The first door prize I have is Lee Arnold. So it looks like she's still with us.
I'm here.
Okay, congratulations. Did you want to tell her what she won, Sarah, or do you want me to just call out the names?
Just call out the names. I will contact you via email to let you know what we have. I have a pair of gloves that's large. I have a moving screen, some soaps, and let's see what else.
All right, then the next one.
Other things.
I'm sorry.
I also have a few other things. Okay, next one.
Next one is Fletcher Wilson.
Okay.
The next one is Keith Raymer, R-A-M-E-R.
Okay.
And the next one is Kent Wicker, W-H-I-C-K-E-R.
Okay.
And there were two more numbers you provided for me, but then we didn't go up that far. So would you like to pick two new numbers? A number between 1 and 17.
Let's have 4 and 12.
All right. The 4 is Kenny Hamilton. And 12 is Tim Falls.
Okay, all right. I will contact you via email to let you choose your prizes, and I will send or bring them by to you. And thank you very much for that. I really appreciate Dr. AMIRI coming and speaking with us. It's nice to have some concrete information about viruses, what they look like, and what we can do about them. So thank you very much. I don't believe we have anything else that we need to talk about. We'll see you on the 7th of November for our next meeting. And I really appreciate you all coming to our meeting. Thanks so much. And Phyllis, we can end the meeting now.
Yeah.
All right.
Thank you. Bye-bye. Bye-bye. Thank you for having me. Yeah.
You're welcome.
Have a great evening. Bye-bye.
Bye.
See y'all.
Bye.